Alzheimer's discovery, a patient and a puzzle. In the early 1900s, a German psychiatrist named Alois Alzheimer encountered a patient who would change the course of medical history. The patient, Auguste Deter, was a 51-year-old woman suffering from a perplexing array of symptoms. Her memory was fading, her thoughts were muddled, and her behavior was becoming increasingly erratic. Dr. Alzheimer was determined to understand the root of Auguste's condition. He meticulously documented her symptoms and, after her death in 1906, performed a groundbreaking autopsy. Examining her brain tissue under a microscope, he discovered something extraordinary: abnormal clumps and tangled fibers. These would later become known as amyloid plaques and neurofibrillary tangles, the hallmarks of the disease that would bear his name. Dr. Alzheimer's findings were met with a mixture of curiosity and skepticism. Some dismissed his observations as a rare anomaly, while others recognized their potential significance. Little did they know that they were witnessing the birth of a new field of study dementia research. The journey to understand and treat dementia had begun, a journey that would span over a century and involve countless brilliant minds across the globe. Section 2. Unraveling the mysteries, plaques and tangles. Dr. Alzheimer's discovery of plaques and tangles in Auguste Deter's brain sparked intense scientific curiosity. Researchers were eager to understand the nature of these unusual structures and their role in the development of dementia. Over the ensuing decades, they painstakingly pieced together the puzzle, uncovering the secrets hidden within these microscopic abnormalities. Plaques, they learned, are sticky deposits of a protein called beta amyloid that accumulate between nerve cells in the brain. These plaques disrupt communication between neurons, interfering with the brain's ability to function properly. Tangles, on the other hand, are twisted fibers found inside neurons. They are formed from a protein called tau, which normally helps to stabilize the internal structure of nerve cells. In Alzheimer's disease, tau becomes abnormally modified, causing it to twist into tangles that disrupt the transport of nutrients and signals within the neuron. As researchers delved deeper into the intricacies of plaques and tangles, they began to understand their devastating impact on the brain. These microscopic structures were not merely bystanders in the progression of dementia. They were key players in the destruction of brain cells and the decline of cognitive function. Section 3. Beyond Alzheimer's, a spectrum of dementia. For many years, Alzheimer's disease was considered a single, homogeneous entity. However, as scientists gained a deeper understanding of the brain and its complexities, they realized that dementia was not a one-size-fits-all diagnosis. It was a spectrum of disorders, each with its own unique characteristics, causes and consequences. Vascular dementia, for example, emerged as a distinct type of dementia caused by damage to the blood vessels in the brain. This damage can be caused by strokes, mini-strokes, or other vascular problems that restrict blood flow to brain cells, depriving them of oxygen and nutrients. Frontotemporal dementia, another type of dementia, was found to primarily affect the frontal and temporal lobes of the brain, areas responsible for personality, behavior, language, and motor skills. Unlike Alzheimer's disease, which typically affects people over the age of 65, frontotemporal dementia often strikes individuals in their 40s and 50s. The realization that dementia encompassed a range of disorders was a crucial step forward in the field. It highlighted the need for more precise diagnostic tools and personalized treatment approaches tailored to the specific type of dementia and its underlying causes. Section 4. A new era of imaging CT scans and neurotransmitters. The latter half of the 20th century witnessed remarkable technological advancements that revolutionized the field of medicine. The advent of computed tomography, or CT scanning, in the 1970s provided doctors with an unprecedented glimpse into the living brain. For the first time, they could visualize the structure of the brain without invasive procedures. CT scans allowed doctors to identify structural abnormalities such as brain atrophy, suggesting the presence of dementia. This non-invasive imaging technique became an invaluable tool for clinicians. Researchers also made significant strides in understanding neurotransmitters in brain function and dysfunction. Neurotransmitters facilitate communication between neurons, and their disruption plays a crucial role in cognitive decline. Acetylcholine, involved in learning and memory, emerged as a promising target for Alzheimer's treatment.